many Antarctic invertebrates that live at the bottom of the ocean are not protected to crabs, for example, that are very common in other areas, but they are not living in these areas for thousands of years. So the chemistry they have would not protect them in case there is invasive crabs coming. And so that's important because with global change, the chances that these crabs arrive are getting higher and higher. And so the organisms there may not be able to be protected against them. So this is something we have to monitor.
I believe the development of fouling release type paints are very important, exciting area, because when we compare this area with the current technology, fouling release type paints release no toxic chemicals to marine ecosystem. On the other hand, the current anti-fouling paints releases very toxic compounds to marine ecosystems. Therefore, I can say that development of fouling release type paints are of great importance.
Yeah, a very exciting development in my research area is our ability to use soil microbes in order to enhance plant responses against herbivores pests.
We are discovering the effect of some microbes in changing the volatile profile of a attacker plant, which can attract natural enemies. And so far, the role of such hidden player was not really evaluated.
So at the moment, what we do in our group is to shift a little bit focus from the impact of plant chemistry onto tiny herbivores, which were insects, to the effect of plant chemistry onto larger herbivores, which are human. So the aim is here to understand how the chemistry from the plant we grow in agriculture ends up into our plates and will affect directly our health through our microbiome or directly. And something that was really exciting is the discovery and the development of a full database that aims at really screening what is present in our food, including what are the plant metabolites which are known to be bioactive, to be present in our plates so that we can better understand the interactions between plant compounds and human health.
We could control aphids with sugar. There are really big problems in apples. Rosy apple aphid is one of the biggest problems in apple. For years we've been spraying neonicotinoids, and neonicotinoids are insecticides that sort of get into the leaves and even into the flowers, and we know what sort of effects it has on pollinators and many other beneficial organisms. The solution is actually quite simple, but you need to study the chemical interactions of the aphid colonies that surround these aphid colonies. And one thing that really struck me is that there's a lot of ants tending to these aphids. And when there are ants around the colonies, the natural enemies can't do their job because they're being chased away or eaten by the ants. In this case, we could divert the ants from tending, because they're interested in the aphids mostly because of the sugars that the aphids excrete. So if we put sugar bait at the base of the tree with some amino acids to really keep them interested, they didn't attend to the aphid colonies and the natural enemies came in.
People are thinking that researchers always every day find some exciting things, but it's not true. I face constant disappointment at my area because I am a field chemical ecologist, which means that I'm trying to test those volatiles at the field, which mostly working in the lab, and I realized that they don't work at the field at the most of the time because there are so many factors outside. Sometimes you face with good results, and recently I started working with butterflies, which are really a shaded area of chemical ecology. I was successful to find some attractant for them, and it was really good experience and motivation to continue my work.
So we have some spectacular findings. We found a new pollination syndrome where plants mimic the alarm pheromones of insects. These insects release while they are eaten by another organism to betray insects that feed on insects while they are eaten by another organism. So it's a highly complex situation. If a spider, for example, or praying mantis collects a honeybee, the honeybee is of course not happy, releases some alarm pheromones while it is eaten by this spider or praying mantis. And the compounds released by these honeybees are used by flies that are known as food stealers. They fly to the honeybee while this honeybee is eaten by another organism, land on the honeybee and feed on secretions of the honeybee and thereby steal the food of the spider or the praying mantis. And we found a plant which releases the compounds that are normally released from a honeybee to attract and deceive these flies as pollinators.
Discovering of invasive species thanks to chemical ecology.
When we developed a pheromone and color trap for the tomato pinworm, the South American tomato pinworm, which is a very big pest in tomato production in sub-Saharan Africa. So what happened was that we used the pheromone of the predator of the tomato pinworm, and then in combination with a different color trap, we could attract both the pest and the predator on the farm. And with that, farmers use less sprays.
A really, I think, interesting development in the research area of chemical communication between organisms is the use of real-time measuring of the chemical cues that are present in the atmosphere and to have a kinetic of this emission and during a certain period of time.